which are all micro-mediated processes, could also be thoughtful as a form of biotechnology. It is used in a restricted sense today to refer to such of those processes which use genetically modified organisms to achieve the same on a larger scale. Further, many other processes techniques are also included under biotechnology. For example, in vitro fertilization, leading to a test tube baby, synthesizing a gene and using it, developing a DNA vaccine, or correcting a defective gene are all parts of biotechnology. The European Federation of Biotechnology (EFB) has a given a definition of biotechnology that encompasses both traditional view and modern molecular biotechnology. The definition given by EFB is as follows: the integration of natural cells and organisms, cells part thereof, and molecular analog for products and services. Principles of biotechnology. Among many, the two core techniques that enable birth of modern biotechnology are genetic engineering techniques to alter the chemistry of the genetic material DNA and RNA to introduce this into host organism and thus change the phenotype of the host organism. Bioprocess engineering, maintenance of sterile microbial contamination free ambience in chemical engineering processes to enable growth of only the desired micro eukaryotic cell in large quantities for the manufacture of biotechnological products like antibiotic, vaccine, enzyme, etc. The advantage of sexual reproduction over asexual reproduction. The former provide opportunities for variation and formulation of unique combination of genetic setup, some of which may be beneficial to the organism as well as the population. Asexual reproduction preserves the genetic information, while the sexual reproduction permits behavior. Traditional hybridization procedures used in plant animal breeding are often used to inclusion and multiplication of undesirable genes along with desired genes. The technique of genetic engineering, which include creation of recombinant DNA, use of genetic cloning and genetic transfer, overcome this limitation and allow us to isolate and introduce only one or a set of desirable genes without introducing undesirable genes into the target organism. Most likely, this piece of DNA would not be able to multiply itself in the progeny phase of the organism, but when it gets integrated into the genome of the recipient, it may multiply and be inherited along with the host DNA. This is because the alien piece of DNA has become part of a chromosome which has the ability to replicate. In a chromosome, there is a specific DNA sequence called the origin of replication, which is responsible for initiating replication. Therefore, the multiplication of any alien piece of DNA in an organism, it needs to be a part of the chromosome which has a specific sequence known as origin of replication. Thus, an alien DNA is linked with origin of replication so that this alien piece of DNA can replicate and multiply itself in the host organism. This can also be called as cloning or making multiple identical copies of any template DNA. The first instance of construction of an artificial recombinant DNA molecule. The construction of the first recombinant DNA emerged from possibility of linking a gene encoding antibiotic resistance with a native plasmid autonomously replicating circular extrachromosomal DNA of Salmonella type. Stanley Cohen and Herbert Boyer accomplished this in 1972 by isolating the antibiotic resistance gene by cutting out a piece of DNA from a plasmid which was responsible for conferring antibiotic resistance. The cutting of DNA at a specific location became possible with the discovery of the so-called molecular seizure restriction enzyme. The cut piece of DNA was then linked with the plasmid DNA. This plasmid DNA acts as vectors to transfer the piece of DNA attached to it. You probably know that the mosquito acts as an insect vector to transport the malarial parasite into human body. In the same way, a plasmid can be used as vector to deliver an alien piece of DNA into the host organism. The linking of antibiotic resistant genes with the plasmid vector become possible with the enzyme DNA ligase, which acts on cut DNA molecules and join their end. This makes a new combination of circular autonomously replicating DNA created in vitro and is known as recombinant DNA. When this DNA is transferred into the Escherichia coli, a bacterium closely related to Salmonella, it would replicate using the new host DNA polymerase enzyme and make multiple copies. The ability to multiply copies of antibiotic resistant gene in E. coli is called cloning of antibiotic resistant gene. In E. coli, you can hence infer that there are three basic steps in genetically modifying an organism: identification of DNA with desirable gene, introduction of the identified DNA into the host, maintenance of the introduce DNA in the host and transfer of the DNA to its progeny. The genetic engineering or recombinant DNA technology can be accomplished only if we have the key tools: restriction enzyme, polymerase enzyme, ligases, vectors, and the host organism. Restriction enzyme. In the year 1963, two enzymes responsible for restricting the growth of bacteriophage in Escherichia coli were isolated. One of these added methyl groups to DNA while the other cut DNA. The latter was called restriction endonuclease. The first restriction endonuclease hint to whose functioning depended on a specific DNA nucleotide sequence isolated and characterized 5 years later. It was found that the hint to always cut DNA molecule at a particular point by recognizing a specific sequence of six base pair. This six specific base sequence is known as a recognized sequence for hint to. Besides hint to, today we know more than 900 restriction enzymes that have been isolated from over 230 strains of bacteria, each of which recognize different recognition sequences. The convention for naming this enzyme is the first letter of the name comes from the genus, and the second two letters come from the species of prokaryotic cell from which they were isolated. E. coli comes from the Escherichia coli RY30. In E. coli, the letter R is derived from the name of the strain. Roman number following the name indicate the order in which the enzymes were isolated from that strain of bacteria. Restriction enzyme belongs to a larger class of enzymes called nucleases. There are two kinds of exonuclease and endonuclease. Exonuclease remove nucleotides from ends of the DNA, whereas the endonuclease make cuts at specific locations within the DNA. Each registered endonuclease function by inspecting the length of the DNA sequence. Once it found its specific recognized sequence, it will bind to DNA and cut each of the two strands of the double helix at a specific point in their sugar phosphate backbone. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence. These are groups of letters that form the same word, and when read both forward and backward, Malayalam as against a word palindrome where the same word is read in both directions. The palindrome is a DNA in DNA is a sequence of base pair that reads same on the two strands, and when orientation of reading kept the same. For example, the following sequence reads the same on the two strands in 5 to 3 days release. Also, so if made in the 3 days, 5 days, GAATTC, restriction enzymes cut the strands of DNA a little away from the center of the palindromic site, but between the on the opposite strand. This leaves single stranded portion at the end. They are overchanging stretches called the sticky ends. On each strand, these are named so because they form hydrogen bonds with their complementary cut paths. This thickness of the end facilitates the action of the enzyme DNA ligase. Restriction endonucleases are used in genetic engineering to form recombinant molecules of DNA which are composed of DNA from different sources genomes. When cut by the same restriction enzyme, the resultant DNA fragments have same kind of sticky ends, and this can be joined together into an using DNA ligases. You may have realized that normally, unless one cuts the vector and the source DNA with the same restriction enzyme, the recombinant vector molecule cannot be separated. Separation and isolation. The cutting of DNA by restriction enzyme results in a fragment DNA. These fragments can be separated by detecting non-gel electrophoresis. Since DNA fragments are negatively charged molecule, they can be separated by forcing them to move towards the anode under an electric field through a medium matrix. Now, the most common used matrix is agarose, which is a natural polymer extracted from seaweeds. The DNA fragments separate resolve according to their size through sieving is provided by the agarose gel. The separated DNA fragment can be resolved only after the staining the DNA with a compound known as lithium bromide followed by exposure to UV radiation. You cannot see pure DNA fragments in the visible light and without staining. You can see bright orange colored bands of DNA in the lithium bromide stain gel exposed to UV light. The separated bands of DNA are cut out from the agarose zone extracted from the gel piece. This step is known as elution. The DNA fragments purified in this way are used in constructing recombinant DNA by joining them with cloning vector. Cloning vector. You know that plasmid bacteriophage has the ability to replicate within the bacteria cell independent of the control of chromosome. DNA bacteriophage, because of their high number percent, have very high copy numbers of genome within the bacterial cell. Some plasmid may have only 1 or 2 copies per cell, whereas other may have 15 to 100 copies per cell. The number can go even higher if we are able to link an elegant piece of DNA with a bacteriophage or plasmid DNA. We can multiply the number equal to the copy number of the plasmid or bacteriophage. Bacteria use the presence and engineering in such a way that they help easy linking of the foreign DNA and selection of recombinant from non-recombinant. The following are the main features that are required to facilitate cloning into a vector. Origin of a replication. This is the sequence from where the replication starts. Any piece of DNA linked to this sequence can be made to replicate within the host cell. This sequence is also also responsible for controlling the copy number of the linked DNA. So, if one wants to recover many copies of the target DNA, it should be cloned in a vector whose origin support high copy number, selectable marker. In addition to ORI, the vector requires a selectable marker which helps in identifying, eliminating non-transformant as selectively permitting the growth of the transformant. Transformation is a procedure through which a piece of DNA is introduced in the host bacteria. Normally, the gene encoding the resistance to antibiotics such as ampicillin, chloramphenicol, tetracycline, or kanamycin are considered useful selectable markers for E. coli. The normal E. coli do not need carry resistance against any of these antibiotic cloning sites. In order to link the alien DNA, the vector needs to be very preferably recognizable site from the commonly used external enzyme. Presence of more than one recognizable site within the vector will generate several fragments which will complicate the gene cloning. The regulation alien DNA is carried out at a resistance presence in one of the two antibiotic resistance. For example, you can get a foreign DNA at the BAM-H1 site of vector tetracycline in the vector PBR322. The recombinant plasmid will lose tetracycline resistance due to insertion of foreign DNA but can still be selected out from non-recombinant one by plating this transformant on the tetracycline containing medium. Transformant growing on ampicillin containing medium are then transferred on a medium containing tetracycle. The recombinant will grow on ampicillin containing medium but not on that containing tetracycle. But non-recombinant will grow in medium containing both the antibiotic. In this case, one resistant antibiotic resistance helps in selecting the transformant, where the other antibiotic resistance is inactivated due to insertion of alien DNA and helps in selection of recombinase. Selection of recombinase due to inactivation of antibiotic is a cumbersome procedure because it requires simultaneous plating on two plates having different antibiotics. Therefore, alternative selective markers have been developed which obtain a recombinant from non-impedant on the basis of their ability to produce color in presence of homogenic substrate. In thus, recombinant DNA is inserted within the coding sequence of the enzyme beta-galactosidase. This results in the inactivation of gene for sensitivity of this enzyme, which is referred to as insertional inactivation. The presence of the chromogenic substrate gives blue-colored colonies of the plasmid in the bacteria does not have an insert presence of insert results into insertional inactivation of the alpha galactosidase and colony do not produce any color. This identifies the recombinant colony vector for cloning genes in plants and animals. You may be surprised to know that we have learned the lesson of transferring genes into plants and animals from bacteria, viruses, which have done this for ages, how to deliver genes to transfer eukaryotic cells and force them to do what the bacteria viruses were. For example, agrobacteria proliferation, a pathogen of several dicos, is able to deliver a piece of DNA, non-HD DNA, to transfer normal plant cells into the tumor and direct this tumor cell to produce the chemical required by the pathogen. Similarly, retrovirus in animals have the ability to transfer normal cells into cancerous cells. Vector understanding of the art of delivering genes by pathogen in their eukaryotic hosts has generated knowledge to transform these tools of pathogen into useful vector for delivering genes of interest to humans. The keyword relation class with agrobacterium has now been modified into cloning vector, which is no more pathogenic to the plant, but is still able to use the mechanism to deliver gene of our interest into variety of plants. Similarly, retrovirus have been disarmed and are now used to deliver disable genes in animal cells. So once a gene or the DNA fragment has replicated in the suitable vector, it is transferred into bacterial plants and animal host where it multiplies. Since DNA is a hydrophilic molecule, it cannot pass through cell. In order to force bacteria to take up the plasmid, the bacteria cell must first made to compete to take up DNA. This is done by treating them with a specific concentration of divalent cations such as calcium, which increases the frequency with which DNA enters the bacterium through pores in its cell wall. Recombinant DNA can then be forced into such cell by incubating the cells with recombinant DNA on ice, followed by pressing them deeply at 42 degrees Celsius heat shock and then putting them back on ice. This enables the bacteria to take up the recombinant DNA. This is not only way to introduce DNA into host cell in a method known as a micro-injection. Recombinant DNA directly injected into the nucleus of an animal cell. In another method suitable for plants, cells are bombarded with high-velocity microparticles of gold or tungsten coated with DNA in a method known as ballistic or gene gun. And the last method uses disarm pathogen vectors which and allowed to infect. We transfer the recombinant DNA into the host. Recombinant DNA technology involves several steps in specific sequences such as isolation of DNA, fragmentation of DNA by restriction, isolation of desired DNA fragment, ligation of DNA fragment into vector, transferring the recombinant DNA into the host, culturing the host cell a medium at large scale extraction of desired product, isolation of genetic material DNA. Nucleic acid is the genetic material of all organisms without exception. Majority of organisms, this is the deoxyribonucleic acid of DNA. In order to cut DNA with the extraction enzyme, it needs to be imbued from other macromolecules. Since the DNA is enclosed within the membrane, we have to break the cell open to release DNA along with other macromolecules such as RNA, protein, polysaccharide, and soluble. This can be achieved by treating the bacteria cells of plant cells, animal tissue such as rhizosome bacteria, cellulose plant cell, chitinous fungus. Genes are located on long molecules of DNA intertwined with proteins such as acetone. RNA can be removed by tetraploid ribonuclease, where the protein can be removed by tetraploid proteins. Other molecules can be removed by appropriate treatments and purified DNA ultimately precipitate out of the digestion of gene addition can be seen in a collection of fine threads in the suspense. Cutting of DNA at a specific location. Restriction enzyme digestion are performed by incubating purified DNA molecules with restriction enzymes at the optimal condition for that specific enzyme. Agarose gel electrophoresis is employed to check the progression of a restriction enzyme digestion. DNA is a negatively charged molecule and it moves toward the positive electrode anode. The process is repeated with the vector DNA also. The joining of DNA involves several processes after having cut the source DNA as well as the vector DNA with a specific restriction enzyme. The cut out gene of interest from the source DNA and the cut vector with space are mixed and ligases. Either this results in preparation of recombinant DNA, amplification of gene of interest using PCR. PCR is experiment polymerase chain. In this reaction, multiple copies of DNA or interest is embedded in vitro using two sets of primers. Small chemically synthesized oligonucleotides that are complementary to the region of DNA and the enzyme DNA polymerase. The enzyme extends the primer using the nucleotide provided in the reaction and generating DNA as template. In the process of replication of DNA repeated many times, the segment of DNA can be amplified to approximately billion times. 1 billion copies are made. Such repeated applications are achieved by use of the thermostable DNA polymerase isolated from Bacterium thermus aquatica, which remain active during the high temperature and induced denaturation of the standard DNA. The amplified fragment, if desired, now can be used to integrate with the vector for further cloning. There are several methods of introducing the legated DNA into the suspense. The suspense after making the competent to receive take up DNA presence in its surroundings. So if a recombinant DNA bearing gene for a restriction to antibiotic ampicillin transformed into E. coli cell, the host cell become transformed into ampicillin resistant cell. If we spread the transport cell on a agar plate and containing ampicillin, only transformant will grow, untransported recipient will die. Since due to ampicillin resistance, one is able to select a transport cell in the presence of ampicillin. The ampicillin resistance in this case is called selectable mark. When you insert the piece of alien DNA into the cloning vector and transport it into bacteria, plants, or animals, the alien gene gets multiplied. In almost all recombinant technology, the ultimate aim is to produce the reservoir protein. There is a need for recombinant DNA to be expressed. The foreign gene gets expressed under appropriate conditions. The expression of foreign gene in the host cell involved understanding many technical data after having cloned the gene of interest and having optimized the conditions to introduce the expression of the target protein, one has to consider producing it on a large scale. If any protein encoding gene is placed in a heterologous host, it is called a recombinant protein. The cell harboring cloned genes of interest may be grown on a small scale in the laboratory. The culture may be used for extracting the desired protein and then modifying it by using different separation techniques. The cell can also be modified to continuous culture system wherein the used medium is drained out from one side while the fresh medium is added from other to maintain the cells in their physiological most active log exponential phase. This type of cultural method produces a larger biomass is leading to higher yields of desired protein. Small volume cultures yield appreciable quantities of products to produce a large quantity of development bioreactors where large volume 101,000 liters of culture can be processed was required. Diverse bioreactors can be thousands of based on raw materials or biological and quantity of the specific product. Individual enzymes affected by using microbial animals or humans. A bioreactor provides the optimal condition for achieving the desired product by providing optimum growth conditions and better pH substrate, salt, vitamin, oxygen. The most common bioreactor is trading type which are shown. A straight tank reactor is usually cylindrical or with a curved base to facilitate the mixing of the reactor content. The star facilitates even mixing and even ability throughout the reactor. Alternatively, air can be bubbled through reactor. If you look at the figure closely, you will see that the bioreactor has an agitator system and engine. oxygen delivery system and a foam control system, a temperature control system, pH control system, sampling post so that the small volumes of the culture can be withdrawn periodic downstream processing. After completion of the biosynthetic stage, the product has to be subjected to a series of processes before it is ready for marketing as a finished product. The processes include transportation and purification, which are collectively referred to as downstreaming processes. The product has to be formulated with suitable preservatives. Such formulation has to undergo through clinical trials, as in case of drugs. Strict quality control testing for each product is also required. Downstream processing and quality control testing vary from product to product. Biotechnology uses a large production, marketing, and products and processes using live organisms or engines. Modern biotechnology used genetically modified organism metabolism only when men learn to alter the chemistry of DNA and construct the recombinant DNA. This key process is called recombinant DNA technology, genetic engineering. This process involves use of artificial nuclear DNA ligase, appropriate plasmid variable to isolate to vary the foreign DNA into host organism, expression of the foreign gene, purification of the gene product, the functioning protein, and finally making suitable formulation for marketing. Large scale production involves use of bioreactors.